And so maybe you've heard, um, Jack and Jill went up the hill, that little nursery rhyme. Okay. Um, no, 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 it wasn't Jack and Jill. It was silly me. I'm forgetting my nursery rhymes. It was Little Miss Muffet, right? And she sat on her tuffet. And what was she doing? She was, I don't know what the hell she was doing. She was like something with curds and whey. I don't know if she was eating the curds and whey or she was separating the curds. Maybe I'm not that good at those nursery rhymes, but I know that Little Miss Muffet, she was doing something with curds and whey. So the curds is what will give rise to cheese. The whey is the liquid part that we don't want. And like I said before, in the past, and it wasn't that long ago in the past, if you wanted to produce cheese, you would have to go and get the enzyme called chymosin, which basically, is able to go and denude casein of the surface sugars in order for it to coagulate. And that chymosin would be coming from the stomach of calves and goats, but baby calves and why babies? Because the baby calf and the baby goat, they're going to be feeding off of mother's milk. And it's at that stage in their life when they're producing a lot of this enzyme. And that's why baby cows and baby goats were targeted, because the adults, they're not messing with milk, they're eating like vegetation. Okay. And so, but now, we don't have to do that. We don't have to kill baby cows and baby goats because we like cheese. Instead, we have biotechnology to help us out. So this over here is in our textbook. And what this shows are the three different ways, the two different processes that we can go and use biotech to go and make as much chymosin enzyme as we need to make as much cheese as we want. So the two different methods here, one is going to go and be doing an mRNA extraction from calf cells. So you would do a little biopsy, okay? It's a one-time thing. It's a one-time thing. You do a little biopsy of maybe, calf stomach, in order to go and find the mRNA transcripts that are going to be translated into chymosin. And then from there, we're going to go and take that mRNA and we're going to go and make cDNA using reverse transcriptase enzyme. And now what we have is we have cDNA of the gene without the introns. And the reason why that's important is because when we go and take this transgene and put it into bacteria to make as much chymosin as we can possibly need, bacteria don't have the ability to splice out introns. So if we went and just took genomic DNA for the chymosin gene and put that in a plasmid and then transform bacteria, we wouldn't be getting functional chymosin because the introns are still there and bacteria do not do post-transcriptional modification. Okay, so this is one way to do it, with getting our mRNA without the introns, making it into cDNA, getting that into a plasmid, and then starting from there. The other way to do it is to figure out what is the amino acid sequence of chymosin. Okay, and there's biochemical ways to do that you could actually figure out what is the amino acid sequence. And once you know the amino acid sequence, you can produce DNA synthetically. So you can, they have, just like they have DNA sequencers, they have DNA synthesizers that are actually putting together nucleotides in a specific order that you require. And so this is just one way of getting DNA that is going to be your transgene of interest, but without the introns. Okay. After that, everything else is pretty much the same. So it's getting that cDNA into an appropriate plasmid vector, introducing the vector to a suitable microorganism, and E. coli works great, Escherichia coli. Then you got to do expression and excretion of that protein in culture. And then what you got to do is you got to go and convert what's called prochymosin into active chymosin. So the protein has got to fold in just the right way. And it does this at low pH. And it makes sense, right? Because if this chymosin is normally active in the stomach, of baby cows and goats, well, we know the stomach is going to be, have a low pH. So what we got to do is we got to go and then take our prochymosin, put it at a low pH in order to get the right conformation. And then what we want to do is purify that protein. And once we do that, now we can go and milk a cow, and we can hit that milk with chymosin, and then we can start to get the curds that form, and then we can, continue the process to make cheese. So uh, proteins are a big part of biotechnology, and not just for cheese making or making of alcoholic beverages, but also really important enzymes, hormones, and antibodies. Okay, enzymes that can be used for, therapeutic proteins, enzymes that can be used for a textile and clothing industry, enzymes that can be used for bioremediation, and enzymes that can be used for food processing. And this is just, just a few of the many, many examples. So this right over here is in our textbook, this little flow chart. And now we're going to move on. So biotech production of target proteins. So proteins can be produced in a whole variety of different cell types. Everything from, bacterial microbes to mammalian cell culture and in between. So what we're going to learn is that we can produce our proteins in bacteria, which are prokaryotic cells. We can do it in very simple eukaryotic cells like yeast, fungi. We can use insect cells. We could use plant cells, and we can also use mammalian cells. Okay. So when do we use? How do we know, when to use which cell type? Well, it really depends on the protein that we're producing, and we'll talk more about that in a little bit. So producing proteins, it is a complicated process, and it is very time consuming. So it's not easy to do, but if you have the right equipment and the right expertise, it is something that can be done on a very large scale. And when we talk about large scale, we're talking about batch amounts. So large batches of the desired protein can be produced in bioreactors that are like the size of like small pools, okay? But of course, not like a swimming pool, but more like, a stainless steel, huge, massive container that can be sterilized, okay? So what is a bioreactor? It is a cell system that produces biomolecules, okay? So if we want to produce proteins, we can do that in a bioreactor. Now, within bioreactors, there's a lot of stuff, a lot of different parameters that need to be strictly maintained within uh, within a very narrow range. And so it all depends on what kind of cells that we're trying to go in and grow. Okay. So if it's, for example, E. coli, well, we really know a lot about E. coli. We know the absolute, optimal temperature for them to grow, the optimal oxygen concentration, the optimal pH. We already talked about temperature, the CO2 concentration, the optimal amount of nutrient in the media that they're growing in. We know all that, but within bioreactors, all of these different parameters that I mentioned, they've got to be monitored very closely, because if the temperature rises too much, or it drops too much, or the pH changes, or the oxygen or CO2 concentrations get out of whack, then that whole entire batch, can be worthless. And that could very well be, tens of thousands of dollars or even more. Okay. So the next step is, after we get all the ideal growing conditions, is that at an appropriate time, the proteins have got to be isolated. So at a particular time in that growth phase when the culture in these huge bioreactors are producing the proteins that they need to be producing, at some point we're going to have to go and harvest those proteins. And it's not that easy because there are other proteins that are going to be produced by the cell, and you got to separate your protein of interest from all the other proteins that the cell normally produces. Okay, and it's a very uh, technologically challenging process, but with the kind of technology and different methods that we use in biotechnology, it can be done, and it can be done efficiently and cost effectively too. Okay. Now, when we're producing proteins, if it's anything that's going to market, anything that's going to market uh, in the form of human consumption or anything of that nature, the whole process is highly regulated. It's regulated by FDA, okay? Uh, it's regulated by USDA, and it's also regulated by EPA. Okay, so FDA, Food and Drug Administration, USDA, U.S. Department of Agriculture, and EPA, Environmental Protection Agency. So, you're not just a company that's just doing this willy nilly to produce proteins, like mad scientists, style. It doesn't work that way. Okay. The whole entire process is heavily regulated, and it's important. It's important for it to be heavily regulated. Okay, so this right here is an example of what a bioreactor would look like. Okay, so it's typically like some kind of stainless steel vessel. Inside, we would have media. We would have what's known as an impeller to make sure that all the components of the media, all the nutrients, are moved about and is circulating. So that way, the cells and the nutrients don't just wind up falling out, in the very bottom of the container. So by making sure that everything is moving about, we can get the optimal growth that we need. So we're going to be measuring oxygen concentration. We have a cooling jacket to go and make sure that we're at optimal temperature. We got temperature probe. We got a pH probe. We have ways that we can adjust pH by adding, small amounts of acid or base. We can monitor pressure. We have a harvest pipe that we can go and harvest the media that is going to have, our cells and the proteins. And then after, when everything's done and we Okay, guys, we should be back in business. Can you guys hear me good now? Okay, excellent. Okay, if you already have your hand up, the little cartoon hand, put it down so that way if something happens again, and everybody puts their hand up, I know that there's a problem. Okay, so if your hand, the little cartoon hand is up, just go and take it down so that way if there really is an issue, then I will be alerted. Okay, so let me go back over here and share. Let's go and continue. We were learning about bioreactors. And so the only, what I was saying is that these bioreactors, all of the parameters that we talked about are strictly maintained. Because if you get a little bit out of whack, if the temperature, goes up a couple degrees or goes down a couple degrees, or the pH jumps, by one or two, I mean, that can totally ruin this batch. And, that could be, tens of thousands of dollars that, you know, that you're just going to have to eliminate. And remember, in this process, time is money. So this whole entire process can take, 3 months. It can take 90 days in order to get the protein that you're trying to produce. And if you got to go and throw out that batch, man, not only are you out money for everything that it takes to get this batch and the media and the cells and everything together, but you're the money that you'd be able to generate by producing your therapeutic protein or your industrial enzyme, that's out the window too. So you have pH probe, temperature probe, impeller to make sure everything is getting moved around and all things like that. So this is pretty neat. This is like a for fun slide, just to have an idea of what the bioreactor is all about. Okay, so over here, this is also a for fun slide. So a for fun slide means that you don't have to remember all these enzymes, but this is just a handful, just a really, really small number of enzymes that are used in industrial applications. Everything from amylases that are going to be digesting starch, different proteases for textile, clothing industry, for food processing, you name it, lipases, okay, breaking down fats, pectinases. How many people like their apple juice or their orange juice or whatever it is? Pectinases are really important because it helps to digest essentially the cell wall of plants in order to be able to get at those juices that are trapped inside the cells. Everything from lactases. There's a lot of people that like milk or like cheese, but it doesn't like them, right? Because it gives you bad indigestion, gas production, messed up stomach, all that. But you can drink milk and dairy, have dairy, if you hit it with lactases that can break down lactose. And so, lactases are important. Glucose isomerase for producing high fructose corn syrups. Yeah, we probably don't need, too much of that, right? That's not like the best thing, high fructose corn syrup, but nonetheless, it's in our foods, and even production of penicillin. Okay, so over here the question is, what additional enzymes studied in chapter 3 would need to be mass produced for recombinant DNA technology? So everything that we learned about with regards to PCR, to reverse transcription PCR, all of the stuff, from, getting our gene into a plasmid vector, from being able to cut DNA at particular areas, based on corresponding sequence. What are some of the proteins, the recombinant DNA technology proteins that we would want to produce using biotech? Can you guys in chat, can you guys think of some of the different enzymes and, which are proteins that we would need to be able to even do, biotech? Let me see what you guys are putting. I got to go and stop the PowerPoint to be able to go and look at the chat. Excellent, excellent, excellent. Yeah, yeah, you guys, you're putting a lot of good stuff. Think about it. Think about it. What about all the different restriction enzymes? Okay, there are hundreds of different restriction enzymes. Okay, all of those need to be mass produced. They all got to be mass produced. And guess what? There are companies that do just that. And I'm trying to see where my book is. Where's my book? Here it is, right here. Okay, this right here is a catalog. This right here is a catalog. Okay, it's called New England Biolabs. Look how thick this catalog is. It's like a freaking book. Okay, what's in here? All the things that you can buy to be able to do genetic engineering. Okay, so of course over here, you start off. See. Okay. Look, right here. Okay, you have pages and pages of different restriction enzymes that you could buy, depending on where you want to go and do your cutting. Okay, look over here. Okay, look at all these enzymes. Look at all these enzymes that you could buy. Okay, now let's take a look at. Let me see if they have the cost here. Sometimes they don't have the cost. You have to go to the website. But you got this whole freaking book here. Okay, so how do you think that they're able to go and supply researchers all throughout the world with all these different enzymes? So from restriction enzymes to polymerases to RNases to proteases, reverse transcriptase. They also carry, plasmid vectors. Okay, in order to do all these different things, you got to have the tools. You got to have the tools to utilize. Okay, so yeah, biotech and bioreactors and production of proteins for all the different purposes that we mentioned. It's huge. It is massive. Okay, and this is one of many companies that are out there. Okay, you got New England Biolabs, you got Thermo Fisher, you got Promega, you got Invitrogen. It's huge. Okay, but there's a demand out there because that's how we do research. Okay, let's go and move on. Okay, so let's go and take a look at another for fun slide. These are some protein-based pharmaceutical products. So basically, we're talking about like therapeutic proteins. So for people that are anemic, you have erythropoietins, okay, to treat cancer, HIV, and things of that nature, okay? You got different interleukins, which are cytokines associated with the immune response, monoclonal antibodies that are huge. They're huge. They're huge for diagnostic purposes. They're huge for being able to go and, for example, target COVID virus. That's monoclonal antibodies. They're used to target cancer cells. I mean, that's huge. You got blood clotting factors, human growth hormone. You got insulin. You got, vaccines like the flu vaccine. Okay, that's a subunit vaccine, and that vaccine is generated using biotechnology, which is essentially just purifying, particular proteins associated like with influenza, and that's what you get vaccinated with. You don't get vaccinated with the actual flu virus or a killed flu virus or attenuated flu virus. The flu vaccines that are out there, they're basically subunit vaccines. So what they do is they figure out, a particular protein on the surface of the influenza virus, and they mass produce that. But of course, the flu, like other viruses, from one year to the next, they take on a lot of mutations, and so the proteins change slightly, and that's why every year you're supposed to get a flu vaccine, so that way, and which different proteins are they putting in the flu vaccine? Well, the ones that the researchers believe that we're most likely to come across. So no, not every flu strain is present in the vaccine, but the ones that we're most likely to come across. Okay, so there you go. So Now we're going to go and continue. So applications of proteins in healthcare. We're going to do a lot of this when we do medical biotechnology. But for example, treatment of damaged cornea. So cornea is basically that part of the eye that is transparent, that allows light to go and pass through the cornea, the aqueous humor, the pupil, the vitreous humor, so that the light can go and hit at the fovea centralis on our retina, okay? And if our cornea is damaged, you know, there's not a whole lot of vasculature associated with the cornea. So everything that's there is like basically by way of diffusion, okay? Because like, and if you look at someone's cornea, like you're not seeing any blood vessels. And so it's really, really hard to go and, you know, to have that damage cornea heal. But we're using biotech to make a difference. So biosynthetic corneas from synthetically cross-linked recombinant human collagen that are being produced by yeast cells. So we're taking the gene that codes for human collagen, we're taking that transgene, getting it in a plasmid vector, transforming yeast cells, and the yeast cells are producing human collagen, and then we can use that in order to go and repair the cornea from people that have issues. Okay, we can also go and use, you know, biotechnology to go and produce like monoclonal antibodies, which are really, really important for diagnostic purposes. So where it says screening molecules associated with disease, well, you're going to hear this several times throughout this course, that diagnostics are huge, okay? The more efficiently and the earlier you can diagnose a particular disease, the better the prognosis will be. So if you can diagnose a cancer when it's the size of like a pea before it even has a chance to metastasize, it can be cured every time. You can just go and remove it, okay? But if you don't get an early diagnosis and that cancer starts to spread before, you know, you get a diagnosis, then that's not good, okay? So, you know, we're using biotech to develop diagnostics to be able to, you know, identify diseases at the earliest stages possible to get the best prognosis and to help people. And a really good example is using monoclonal antibodies to be able to detect prostate-specific antigen levels in males. So PSA, prostate-specific antigen, it is something that's found, you know, it can be detected in a male's blood. And, you know, PSA levels should be monitored as you get older, okay? And it's always good to have baseline levels. So probably most of the people that are taking this course are under the age of 30, but you would want to go and talk with your doctor, your physician, after when you start to get into your 30s about just getting a baseline PSA test, especially if there's any history of prostate cancer, you know, in males in your family, okay? And if that is the case, then, you know, it might be a good idea just to get baseline information. Don't wait until you have issues with your prostate. But when you start to get in your 30s and you talk with your physician, you know, it might be worth to go and get that PSA level so that way you know how things are when everything is all good, because maybe your levels are just normally high. And that's just you. And it doesn't mean that you have prostate cancer, but nonetheless, you know, so if you have your baseline information, then when you are in your 40s and you do start to experience issues with prostate, if that is the case, then you know at least what your baseline level was, okay? And it's very effective in diagnosing, you know, risk factors associated with prostate cancer. Okay, so now what we're gonna do is take a look at applications of protein in industry. We talked about using different enzymes and proteins for food processing, for, you know, clothing, textiles, leather goods, for detergents, right? Detergents are a big thing. It wasn't that long ago when many of the detergents that we're using were basically, you know, utilizing lots of inorganic phosphates. And yeah, it does a really good job at making your clothes clean, but when the dirty water leaves your washing machine, and if that winds up in the environment, like in aquatic wetlands, those phosphates can be very problematic. You can get eutrophication, where you get, nutrient-rich waters, and you get these algal blooms, and it's really, really bad. But nowadays, many detergents are actually they're actually proteins, they're actually enzymes, and these proteins, just like proteins in our body, they have a lifespan, and they can get easily broken down. So it's dramatically, it's much better for the environment as opposed to using like inorganic phosphates and stuff like that. And also bioremediation. What about cleaning up oil spills and hydrocarbons that leak out from oil tankers and stuff like that? What's the best way to go and clean up an oil spill? Well, it's going to be the way that it has the least detrimental impact on the environment. You could get rid of the oil and the beach might look clean, but you don't want the cure to be worse than the actual problem, right? And some of these, methods used in the past to rid oil from beaches, it would have an effect on the ecosystem. But on the other hand, if you're using, proteins and enzymes that, are capable of breaking down hydrocarbons that are then, easily broken down themselves, then that's something that is going to be better for the environment than using inorganics that are going to wind up within the ecosystem and cause serious problems. Okay, so what I want to do right now is I want to show you a video, and this video is in modules, but so you can watch this later on your own, but I think it's a cool video to see. So let me go and do that.
What do bread, cheese, and soy sauce have in common? They are diet staples of billions of people around the world, and they are produced by fermentation. These foods and many others are made possible by the use of enzymes in food production. Hi, I'm Dr.
Okay, guys, before I go any further, can you hear the video? Can you hear it? Okay, I just want to make sure you can hear it. Okay, so I'm going to go and continue.
Dr. David Targan, and welcome to BioBites. So what exactly is fermentation? Well, it's the conversion of the carbohydrates in milk, meat, fish, vegetables, cereals, and other organic matter by microorganisms or enzymes into acids or alcohols. Milk, without refrigeration, will spoil very quickly. But if you turn that same milk into a hard cheese, it can last for many months. This is particularly important in developing nations that lack or are limited in refrigeration capabilities. And it is one reason why fermented foods make up about 1/3 of the diet of humans today. People have been preserving food with this process for thousands of years. So biotechnology is really just inventing new ways to improve this old process. Researchers are now utilizing and modifying enzymes to make fermentation more efficient and also to increase the quality, safety, and product consistency of preserved foods. Enzymes aid in the manufacture of fruit juice, corn syrup, candies with soft centers, beer, cheese, sausage, and many other foods and drinks. In fact, food producers use over 55 different enzymes to make many basic elements of our diets. Biotechnology also makes food enzymes more widely available. Chymosin, an enzyme used to make various cheeses, occurs naturally in the stomachs of calves, lambs, and baby goats. By modifying the genes of microorganisms, scientists have now created a way for yeasts, molds, and bacteria to produce chymosin, eliminating the reliance on livestock for this enzyme. Scientists also are improving the production capabilities of the food industry as a whole. One bacterium used in the production of
Okay, I'm seeing a lot of hands up. Yeah, I know it's slow. The guy's talking like something's wrong with him. Okay, but it has something to do with the connectivity. So if you watch this on your own computer, it shouldn't sound that way. The only thing I can think of is that there's one setting that might make the difference. But I want to make sure that you can still hear, right? Well, let me try. Let me see if I change the setting. Okay? So let me go over here.
many yogurts and cheeses is vulnerable to viral infection. Using recombinant DNA technology, researchers have made some strains of these bacteria resistant to viruses. This means that fewer batches of cheese have to be thrown away due to contamination, resulting in abundant, healthful, and less expensive food for people worldwide. For BioBytes, I'm Dr. David Targan.
Okay. Okay, that last part, could you still hear the video? Okay, so you can still hear it. Okay, great. Okay, and you can hear me fine? You can hear me good? Okay, I just want to make sure. Okay, so yeah, it took like 5 minutes for him to say the 2 minutes that he needed to say, but I don't know. I guess my computer's acting a little bit squirrely today. I'll have to go in and, free up some space on my computer or something. I don't know what the deal is. But nonetheless, let's go and continue. But I thought that was interesting, okay? So just like you said, with food processing, we're using over 55 different freaking enzymes to come up with the normal foods that we find, in our supermarket. And that's something that's pretty amazing. Okay, so let's go and continue. Okay, so now what we're going to do is we're going to learn about something cool. So we know that by using biotechnology, we can make very large quantities of a particular protein that we're interested in, okay? But did you ever think about how maybe we can take an enzyme, a protein, and even make it more efficient at doing what it does, okay? And that is a goal of many people in biotechnology. Now, let me give you the example here, okay? So, a big problem that we have in marine environments is going to be, oil spills, okay? Oil that just leaks out of ships and things of that nature, and, trying to do something about it, right? We would think that the best, one of the easiest fixes is just to go and design better ships and you reinforce the hull so that it doesn't happen, right? But I'm not an engineer and I don't build those ships, so I don't know what the deal is. But nonetheless, I mean, oil still winds up in the water. So instead of using industrial chemicals to break up the oil spill, there are many researchers that are studying different microbes that are naturally present in marine aquatic habitats that can break down hydrocarbons. There are actually microbes that can do it, okay? But they just don't happen to be in great enough numbers to have a really, really big effect. but there are microbes that actually can, break down hydrocarbons and use that as an energy source. So if you're interested in biotechnology, then one of the things that you might be interested in is to figure out what is that biochemical pathway for these particular microbes and bacteria that have these biochemical pathways to break down the oil, to break down the hydrocarbons. And once you figure out what enzymes are at play, the next thing might be is how can we make the enzyme more efficient? How can we make the protein more efficient so that way, we have even better results with cleaning up oil spills, utilizing, these proteins. And so one of the ways that we can do this is through experimentation, and the process is known as directed molecular evolution. Okay, so we know biotechnology is able to produce, different proteins by looking at a particular transgene, but it's also able to design and build new proteins that can even be more efficient than the proteins that are naturally produced. Okay, so invent proteins that are tailored for specific applications. So what directed molecular evolution is all about, it's like speeding up the normal evolutionary process. Okay, so like here's something that that many people agree. So there's lots and lots of plastics that are in the environment, and over time there's going to be microbes that are going to be able to utilize those plastics over time. It might be over hundreds and hundreds of years, but there's going to come like a point where there's so much plastic in the environment that organisms that are able to utilize the components of plastic, the molecular makeup of plastic, that they're going to be able to use it as an energy source. Okay, but who knows? That could be 500, that could be 1,000 years. Okay, but eventually, by way of mutation, there are going to be some organisms that are going to be able to capitalize on all that plastic out there. But that's not our goal, right? Our goal is not waiting that long to do something about plastics. It's either utilizing something other than plastics that is able to get the job done, that is more environmentally friendly and sustainable and things of that nature. But in the example that I gave just a little bit ago is with the oil spill and the hydrocarbons. Okay, so if you're able to isolate marine bacteria that naturally have biochemical pathways to break down hydrocarbons, what we'd want to do is take those genes that are involved in the breakdown of the hydrocarbons, the enzymes, and see, can we make them better without having to wait 100 years and let the process occur naturally. Okay, so that's directed molecular evolution. So it's a method used in protein engineering that mimics the process of natural selection to evolve proteins and nucleic acids towards a user-defined goal. So let's go and take a look at how we would do this. So it starts off, we're going to do our directed molecular evolution by utilizing something known as mutagenic PCR. Okay, so what we're going to do is we're going to go and take that gene that we know is involved in the breakdown of hydrocarbons, because it results in the production of a protein that can break down hydrocarbons. And so what we would do is we can go and take that cDNA, and what we can do is we could do something called mutagenic PCR. So we can go and amplify that cDNA, okay, which came from mRNA. So we've already got rid of the introns, and we can do mutagenic PCR. Now, most of the time when we do PCR, we want the PCR to be high fidelity. We don't want any errors to be made. We want all of the PCR product to be clonal identical. But with mutagenic PCR, we're intentionally wanting the polymerase to make errors, okay? And so there's different ways that we can achieve that. We can go and do mutagenic PCR by utilizing unequal number of the different nucleotides. So by having many more G's than C's in our PCR mix, or many more A's than T's, that can throw things off. We could also increase our magnesium chloride concentration, because remember, magnesium is a cofactor for Taq polymerase. And so remember we said if you add too little magnesium, then the PCR process just is too slow. If you add too much magnesium, then the Taq polymerase goes too fast and is prone to making errors at the expense of moving very fast. So what you could do is, essentially you could add more magnesium chloride to your PCR reaction, and then you have a greater chance at making these errors as your PCR product is produced. So at that point, now instead of having everything, your PCR all clonal, now you're going to have some PCR product that has this mutation, some PCR product that has that mutation, and so forth. And so now what we would do is take that PCR product, and we're going to go and insert that PCR product into a plasmid vector. And then we take that plasmid vector and then we transform bacteria, and we see how successful the bacteria are at being able to break down hydrocarbons. And the ones that are most successful, we keep those and we continue the process. And we keep on doing our mutagenic PCR until we get to a point that it doesn't look like we can increase the efficiency. And at that point, we're not done. Then we can do something called site-directed mutagenesis. And what site-directed mutagenesis is actually creating mutations in different areas of that gene that we think will be able to increase the efficiency. So after, when we do our mutagenic PCR and we get the protein that's produced to be as efficient as possible, we can then sequence that DNA, and we can learn what is the amino acid sequence of that protein, and we can take that information and put it into a special software package that can then figure out, is there any other thing that can be done that we can make this protein more efficient? And maybe it might be a tailored mutation. It might be, well, if we take this particular nucleotide right here and change it so that way we get this amino acid instead of that is the one thing that we can do to make it even that much better. And so when we talk about directed molecular evolution, that is involved with mutagenic PCR, and that's the first step. The next step would be using site-directed mutagenesis to be able to pinpoint the mutations that we need in order to go and get that to be the most efficient protein that we can, okay? A really good example of this was a company that was able to produce bacteria and industrial enzymes that tolerate a cyanide concentration that is normally too high for most bacteria to survive. So let me go and explain why they would do that. Okay, here's the example. You got a family. They saved up all their lives, the whole entire family, to buy 50 acres of land, okay? And why? Because they want to put their house there. They want to have a little farm. They want to whatever. But, maybe you got a bunch of brothers and sisters and this and that, and they all wanted to put in their money to buy this big, 50-acre plot of land. They go and they buy the land only to find out after they purchase the land that it's not land that you can live on. And the reason why is because there's a very high cyanide concentration in the soil, okay? And so, like, what do these people do? And why is there a high cyanide concentration? Well, because they found out after the fact that land during the early 1900s was an agricultural field. And in the early 1900s, many of the pesticides that they utilized had a high cyanide concentration. And over the many, many, many years of applying the pesticide, well, that cyanide built up in the soil. So what do? You could go and, pay a crazy amount of money more than the cost of the land to remove the, the upper, 3 feet of soil with backhoes. But then, where's all that cyanide going? Now you're making it somebody else's problem. And so what do? Well, there's biotech companies out there that, try to solve these kinds of problems. And so there are microbes that are able to actually render cyanide as non-toxic, okay? But the thing is and they can do it naturally. So the whole idea is how can we make that process more efficient? And what happens if we have cyanide levels in the soil that are even too high for the bacteria that naturally occur to be able to survive? Well, then that's when you do your directed molecular evolution. You would go and take the enzymes that are responsible for that biochemical process to render the cyanide as non-toxic. And what you want to do is make the enzymes more efficient. And how do you know if the enzymes are more efficient? Because each time you do your directed molecular evolution, you're going to go and take the bacteria that now have these plasmids with the different mutagenic PCR product, and you're going to plate those bacteria in a media that has a higher concentration of cyanide. And the ones that survive and live, they're able to survive because they're able to render that cyanide, those levels, as non-toxic. And you repeat over and over and over again until you get the enzymes as efficient as possible. And then at that point, you can then go and use those enzymes or microbes in the soil to basically do bioremediation, to make that land, livable again. And that's an example. Okay, so let's go and continue over here. So a company was able to produce bacteria and industrial enzymes that tolerate a cyanide concentration that is normally too high for most bacteria to survive. Okay, and why do we want them to survive? Because we want them to be able to go and render that cyanide non-toxic. Okay, so we're going to go and continue. This right over here is a little diagram, and what this diagram shows is how the process would work. Okay, so for here, you can start off, if you follow my arrow, this is our gene that we isolated, okay? And this gene is responsible maybe for producing a protein that renders cyanide non-toxic, okay? But we want to go and increase the efficiency of the enzyme. So what we're going to do is we're going to go and take the gene, like for example, in the form of like cDNA, and we're going to do mutagenic PCR. And what we're going to get is a whole bunch of different PCR products, that are going to have mutations in many, many different areas, in that cDNA. Now what we're going to do is we're going to take all these different, DNAs that are not clonal, and we're going to go and put it into a plasmid vector, and then we're going to go and transform bacteria like E. coli. And then once we've transformed the bacteria, we're going to perform our experiment. And how do we perform the experiment? Well, if this, for example, is the gene that's going to render cyanide non-toxic, then we're going to go and increase the cyanide concentration in our growth media. And the bacteria that don't survive, well, we're not interested in them. But the ones that are able to grow on that media, we're going to go and culture them. And we're going to go and find out what plasmid they have inside. We're going to go and harvest the plasmid. And then we're going to go and use that insert, that transgene in the plasmid, to undergo another round of mutagenic PCR. And it's cyclic, and we do over and over and over again until we increase the efficiency to a point where we can't get it any better. And then from that aspect, we can then sequence that DNA. We can learn what is the amino acid sequence, and then we can put that amino acid sequence into a protein simulation software package to get the structure of the protein, and then figure out maybe what specific amino acids can we alter or switch out to get any increase in efficiency? And then you would do that by the site-directed mutagenesis. Okay, and then over here, this is in our textbook, and basically, you know, each filter is basically filtering out the best from what you've got, and then doing your directed molecular evolution, and then, you know, putting them through another filter. And what they mean by filter is really, you know, an experimental process to figure out, you know, which is the most efficient. And then that goes to the next round of your directed molecular evolution. Okay, so there we go. Let's go and continue. So now we're probably beginning to understand how important proteins are in terms of therapeutic proteins for diseases that humans have and our domestic livestock and things of that nature, proteins for bioremediation, proteins for the clothing, textile, leather industry, for industrial purposes, for biotechnology. I mean, you name it, okay? So what we're going to learn about now is how do we even start the process of producing a protein of interest, okay? So what we're going to learn is that the whole entire process has two major phases, and they're known as upstream processing and downstream processing. So upstream processing has everything to do with the actual expression of your protein of interest in whatever cells you're going to use, okay? So having, you know, your particular cells that you've chosen to produce your protein of interest in a bioreactor or fermenter, that is your upstream processing. So includes the actual expression of the protein from the cells. Okay, that's upstream. Downstream is what happens after your cell culture produces those proteins. After the proteins are produced, the next step is downstream processing, which is how we're going to isolate and purify those proteins, how we're going to isolate the proteins from all of the other biomolecules, okay? Like, we're not interested in carbohydrate, we're not interested in nucleic acid, we're not interested in lipid, we're interested in the protein. And once you are able to separate proteins from the other biomolecules, how do we separate our protein of interest from all the other contaminating proteins? All of that is downstream processing. So upstream has everything to do with the cells you choose to grow your proteins and the production of the proteins, and downstream has everything to do with purification and isolating your protein of interest. And so it's not, you know, too hard to see that the choices you make during your upstream processing can have an effect on your downstream processing. So there are some things that we can do during upstream processing to make our lives easier or simplify our downstream processing. Okay, so there we go. Let's go and continue. So now what we're going to do is we're going to focus on upstream processing. What we're going to do is learn about the different kinds of cells that we could utilize for protein production, and what are the pros and cons with these different cells. So the first one that we're going to do is bacteria, like, for example, E. coli. So what's so good about using bacteria? Well, we know the optimal conditions to grow E. coli, okay? And the fermentation process is very, very well understood. We can culture huge amounts of E. coli in a very short period of time because they have a 15 to 20 minute generation time. So we can grow huge quantities of E. coli that we're going to use as little factories to produce our protein of interest, okay? They are relatively easy to genetically alter, and we can increase the level of production of a bacterial protein by, inserting additional copies of our gene of interest, or by using a plasmid vector that has a high copy number. Okay, so that's something that's really good. And the most common bacterial species that we use to produce our proteins is E. coli. Okay, so let's take a look over here. Let's learn a little bit more about E. coli and the pros and cons with using bacteria to produce proteins of interest. So bacteria, majority of proteins synthesized naturally by E. coli are intracellular, meaning that the bacteria produce the protein, and the protein stays within inside the cell, and it can often be found in inclusion bodies. Okay, so these inclusion bodies are these insoluble clumps of our protein of interest. Now, the problem with that is because E. coli is not secreting the proteins outside of the cells, and instead the proteins are sticking around inside of the cells. In order to get at our proteins, we got to pop those cells open. And when we pop those cells open, now it makes our lives more difficult, because now we got to separate the protein from all the other biomolecules from within those bacteria. And then we got to separate our protein from all the other contaminating proteins that the bacteria normally produce. Okay? So E. coli can be genetically engineered to produce the desired protein in the form of a fusion protein. Now, I don't know, how many of you have heard of a fusion protein? Raise your hand if you know what a fusion protein is. Anybody? Maybe if you've taken biochem, you know about a fusion protein. So a fusion protein is going to be your protein of interest that is connected to another protein. And then you might be saying, well, like, why would we want to do that? Why would we want to go and take our protein and connect it covalently by way of a peptide bond to another protein? Why would we want to do that? Well, the reason why is because we're smart. So the protein that we're going to connect to our protein of interest is we're going to use it as a way to go and isolate our protein. So we can connect our protein of interest to an enzyme that might target a particular sugar. And then what we can do is we can make a column, an affinity column, they call it. And the purpose of an affinity column is to go and separate your protein from other proteins. So if you line the affinity column with the sugar that particular enzyme that is attached to our protein of interest is going to go and stick onto, then all the other proteins are going to flow out of the column. And what we're left with is our protein of interest stuck to the column. And then there are very, very interesting ways that you can then separate your protein of interest from the protein that is fused to your protein of interest. There are very easy ways to do that. And so, that's something that can be utilized. Okay, so where it says over here, E. coli can be genetically engineered to produce our desired protein in the form of a fusion protein. Why we want to do that? Because this fused bacterial protein is usually an enzyme that will bind to its substrate, like in that affinity column, and then will attach to the affinity column, which is a purification column, and all the other proteins will end up leaving the column because they're not sticking to nothing. And then there are ways that we can go and separate our protein of interest from that enzyme that we utilize to help isolate our protein. Okay, so what are some of the advantages? Well, E. coli genetics are very well understood. E. coli genome is pretty small. It's about like 4 million base pairs, and we understand the genetics really well. We can get an almost unlimited quantity of proteins. We can generate as much protein as we want utilizing E. coli. And the reason for that is that the fermentation technology, meaning that all the growth conditions and the media and the nutrients necessary, is very well understood. What are some of the disadvantages? Well, like we already said, that when E. coli produce proteins, it's not going to be secreted. It stays within the cell, within these inclusion bodies, and we're going to have to pop the cells in order to get our protein of interest out. The other thing is that bacteria do not utilize the same post-translational modifications that eukaryotic cells do, okay? So remember, those post-translational modifications, whether it's glycosylation, acetylation, hydroxylation, methylation, that can have an effect on how the protein is able to fold. And if you don't have the right post-translational modifications, the protein might not fold the way we need it to fold. And that's something to take into consideration. Some proteins are going to be inactive in humans for that particular reason, because the post-translational modifications are not the same as within eukaryotic cells, and as a result, the structure of the protein is not going to be effective for doing the job it's supposed to get done. Okay, so let's go on to continuing with bacteria. So this is our upstream processing. So genetically engineered bacteria can be grown in large-scale fermenters. And why do we call it a fermenter as opposed to a bioreactor? Well, fermenters are, conditions that the bacteria are grown in anaerobic conditions without oxygen, and bioreactors are bacteria that are grown under aerobic conditions. And we already know a lot about bioreactors, and the same thing would go with the fermenter. It's very sophisticated. The computer is going to be monitoring all different parameters necessary for successful growth of our microbes. And the other thing that's interesting, and a lot of people don't know, is that when you're utilizing a bioreactor or a fermenter, you don't want the microbes to produce your protein from the get-go. You don't want those microbes to immediately start producing your protein. Why? Why don't we want that? Why don't we want the bacteria, as soon as we put them in that growth media, to start producing the protein of interest? Why don't we want for that to occur? Can you guys think of a reason why? And maybe you can put that in chat. Anybody? Why don't we want the bacteria to immediately start producing our proteins in the bioreactor or fermenter? Why might we want to wait? Who has an idea? Anybody? I got one person. Okay. Obtain nutrients, make sure conditions are perfect. Okay. Well, it turns out that before we want the bacteria to produce our proteins, we want to make sure that they're doing as best as they could do. Okay, because if they're producing our proteins, then that means that they're expending energy to produce our protein. So we want to make sure that they have everything that they need, and we want to make sure that when we have them to start, when we basically order them or initiate protein production for our proteins of interest, we want to make sure that their growth stage is exponential. That's what we want. We don't want them to start producing proteins before they get into exponential growth. We want them to be into exponential growth. And then that means that, man, they're doing super good. They got everything they need to take care of themselves. Now we can go and start to have them produce our protein of interest. Okay? And then now the question is, how do we do that? How do we order them to start producing our protein of interest? Can you guys think of something that we've already learned about, of how you can be, very, very crafty, that you can be very ingenious in developing a way for the bacteria to produce the protein when you want, exactly when you want. what can we do based on something that we've already learned about? How, yes, 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 yes. Give them lactose or a nutrient that activates the gene of interest. Operator, that's exactly it. Okay? So what you can do is you can go, we know about the lac operon. We know that in front of the lac operon, there's a promoter region, and that we know that the lac operon is only transcribed when there is no glucose and you have the presence of lactose. That's when it goes down. So absolutely, what we could do is we could go and put the lac promoter in front of our gene of interest, and we can go and feed those bacteria glucose. And then when we see exponential growth, we can then stop feeding them glucose and only provide them lactose. Okay? And another way to do that is by offering them, and I don't mean offering them, but putting in that bioreactor a chemical that is known as IPTG. IPTG. And what is IPTG? It is an analog of lactose that doesn't get broken down. And so, if you have your lac promoter and you've got your repressor protein attached, if you add IPTG, it will bind to the repressor, release the repressor, and then you can start producing your protein of interest. You see? So there you go. Learning about something as simple as the lac operon with regards to, how do bacteria regulate gene expression, we can then use that for biotechnology in order to have these bacteria produce our protein of interest when we think is the best time for it to be produced. Okay? So let's go back over here fast. I know that we are super running out of time. Here it is. IPTG binds to lac repressor without being degraded. Okay? And of course, you want to make sure that you're producing, or the bacteria are producing, our gene of interest only when the organism has everything that they need to grow exponentially. Okay? So with that, let me go and see what we have next. Okay, we don't have time to get into the next cell type, which is going to be fungi. So that is where we're going to stop. We're going to stop with upstream processing with bacteria. But you do know the difference between upstream and downstream processing. Upstream has everything to do with the production of the protein, and downstream has everything to do with the purification of that protein that was produced. And we also know pros and cons of using E. coli for protein production purposes. So there you go. Right now, that's our cutoff. That's our cutoff for exam number 2. I'm going to go in the next 10 minutes and put this lecture, publish it in modules. So that way you'll have everything you need if you need to go back or want to go back.